Hello students, I hope you are doing well. So this is the third chapter, nuclear structure and function, and we will be learning a lot of things in this chapter. So these two are the recommended books you can go through on your own. So in this chapter the contents we will be seeing is that cell types according to nuclear arrangement and chromosomal structure, function and organization, and some epigenetic stuff such as histone modification or DNA modification, and how those modifications regulate gene expression, cell cycle and its regulators, cyclins, CDKs, P53, etc. Those regulator proteins of cell cycle, cell division in details, mitosis and meiosis, and errors in mitosis and meiosis, how those errors results in chromosomal abnormalities, and lastly some chromosomal disease that results from chromosomal aberrations such as structural variations and aneuploidy. So moving to the next slide. So here are a figure that depicts the difference of nuclear organization between eukaryote and prokaryote. So we know that eukaryotes nucleus is organized and well defined, whereas the prokaryotes nucleus is not that well defined or not that compartmentalized, that is known as nucleoid. But in the eukaryotes the DNA is well defined within the double membrane and with such a compact space of DNA. So that's the main difference. Although there are some other differences such as cell wall, flagella, but those are not important in this chapter. In this chapter we'll be interested in this nucleus or nuclear structure or nuclear organization of these two types of cells. So moving to the next slide. So here are two figures of one is eukaryotes and other is prokaryote. So the basic difference between these two, we know that from the first chapter that prokaryotes have their DNA or chromosome that is circular, but in the eukaryotes like us we have chromosome that is linear, that is long and linear but not circular. So that's the main difference. And sometimes prokaryotes have plasmid that is just one single DNA that is circular, that is called plasmid. Going to the next slide. So here are some basic differences of chromosomal structure differences that we already know, that the prokaryotes have the circular DNA, whereas the eukaryotes contains a linear DNA molecule, and that linear DNA molecule is compacted in chromatin, that is within the histone proteins. That in eukaryotes their DNA is wrapped around or compacted in histone proteins that give rise to chromatin. And in the metaphase we see the chromosomes, actually the well-known X-shaped chromosomes in metaphase when the cells are active in dividing. So the another difference is naked, that means the DNA is empty, that means only it is associated with only DNA but no proteins are associated, whereas the eukaryotes we see proteins such as histone proteins and other stuffs that is associated with the structure or organization of DNA. We will be seeing that in details. Plasmids are often present, yes, I mentioned that, but in eukaryotes there are usually no plasmid. Plasmid means one circular DNA. And in the prokaryotes there's one chromosome only, but eukaryotes have more than one. Like us, we human have total 46 chromosomes or 23 homologous pair chromosomes. So moving to the next slide. So I will not go in details of this, the structure of the nucleus or function of the nucleus, because we already know we have some idea about the nuclear structure. For example, the nucleolus, nuclear pore, nuclear double membrane, what their function. We already know their function, but I will just like to emphasize few things such as the nucleoplasm that we haven't talked about, and nuclear envelope that it contains two layers. You know that the inner layer and the outer layer. So these two layers have some specific function and structure. So the nuclear envelope that is made up of lipid bilayer, inner layer, outer layer. So inner layer that is inside the nucleus, inner layer is associated with some proteins, special proteins that are known as lamins or nuclear lamins. You can see nuclear lamins, they are made up of filaments which provide the structural support inside the nucleus. That means it helps maintaining the shapes of the nuclear envelope, and it also, the lamins also have a special function in organizing the chromatin. Like lamins are attached to the inner layer, that means in this portion, so it holds chromatins, and when needed it can interact with the chromatin. And we know the nuclear envelope consists of some pores which control the movements between especially mRNA and transcription factors. Those can go in and out. Actually, mRNA can go out, but transcription factors can go in. So depending on the situation. So the nuclear pore allows the transport of those biomolecules. So nucleolus, we know that it is involved in ribosomal RNA synthesis, and that is important for ribosome production. We know that ribosome is made up of rRNA and proteins. Chromatin, we have some new concepts. That is, chromatin is of two types depending on its state. So euchromatin and heterochromatin. So euchromatin are loosely packed and heterochromatin is tightly packed. And tightly packed chromatins do not allow transcription. That means the DNAs are tightly packed, so RNA polymerase and transcription factors cannot bind to those tightly packed DNAs. So heterochromatin or tightly packed chromatin is uh, is not able to undergo transcription or DNA replication. But euchromatin is loosely packed. So we'll be seeing that what makes a chromatin loosely packed or what makes a chromatin tightly packed in details. So the new things is here is the two types of chromatin, euchromatin and heterochromatin. And the lamins, the nuclear lamins, they have very special function in when the cell divides in cell starts to divide and progress, the nuclear structure starts to break down, the envelope starts to break down, then those nuclear lamins helps to dissolve the nuclear structure, nuclear envelope precisely. So going to the next slide. So we all are familiar with this. This shape of the chromosome, that is this X shape. That can be only visible in the metaphase, prophase of metaphase, especially metaphase when the chromosomes line up in the metaphase plate. So this structure is highly condensed, but usually in the other state, when the cell is not under cell division, that means when the cell is growing phase or under resting phase, then this chromosome is not visible. Only this chromatin, these chromosomes are visible in this shape, but just only under mitosis or under metaphase, this X well-known chromosome shape is visible in this shape. But all the other times of a cell's life, this chromatin or DNA or chromosome is like this chromatin, this shape. So here the chromatin is seen in the nucleus during interphase. Interphase I have talked about. Will be seeing the cycle of the cell cycle, the interphase and the mitotic phase. So here is the structure of the chromatin or chromosome. Actually, when these DNA materials are in this loosely shaped or in this not that compact structure, that is known as the chromatin. But when they are very highly condensed structure and this X shape, when the DNAs have undergone the replication process, when the sister chromatids are present, then this X shape well known compacted chromosome structure is visible under microscope, especially in the metaphase phase of the mitosis or massive. So going to the next slide. So here is the figure of a labeled parts of a chromosome. So just note that in the left side this is one chromatid or one chromosome. So when it is single, the chromatids are single, that means it is one chromosome, but when they are attached, that means when the sister chromatids are attached, and they from this structure, these two chromatids are known as one chromosome, sister chromatids together. So both are one chromosome, depending on the state of the cell cycle. So usually the typical chromosome have, or all the chromosome have two arms. The short arm is known as the P arm and the long arm is known as Q arm. So we'll be seeing different length of these arms depending on this. Uh, we'll be seeing the different chromosomes shape depending on this arm, the length of these two arms. And these two arms are connected with the slightly narrow or shrinked region that is known as centromere. So these two arms are joined in this region, centromere, and the end of a chromosome, the two end, it is highly condensed region that is known as the telomere, telomeres end. So know the terms or the regions, it will help you understand further. And also one important thing is the special protein kinetochore. So kinetochore is attached to the centromere. Just know that kinetochore is attached to the centromere, and kinetochore helps spindle binding. So spindle in cell division, when the spindle gets activated, then the spindle binds to the kinetochores. And kinetochores is attached to the centromere. So just know the terms, the kinetochore and the centromere. That's very important. And when the DNA or chromosome has undergone the replication process, when they are duplicated and aligned in the metaphase place and in this well-known structure, then these two chromatids are known as sister chromatids, but together they are known as one chromosome. So chromosome number varies during different cycles of the cell.